This is a study that's bringing together a number of different disciplines, including quantitative genetic interaction mapping, integrative structural modeling, and computational approaches to facilitate merging these different approaches together.
Our lab has been a pioneer in this high throughput mapping of genetic interactions. And since 2005, 15 years ago, we're using something called genetic interactions as a new method for figuring out the structures of protein complexes. And it's been a long road to get from where we started to this.
The technology, both the experimental and the computational technology, has caught up with our vision.
The approach that we show in the paper, where we use this genetic interaction data, it's very complementary to other methods that are available nowadays, and really expand our tool set to understand the structure of protein assemblies and to learn new biology.
In yeast, our readout is colony size. So we basically just measure cell growth, and from that we go back to protein complex structure. And I mean, aside from being really cool that we can do that, the main point there is that we're going from function, pure function, and going back to structure from that. So we're getting a structure that's based on what it actually does in the living cell. So we're adding a layer of making sure that the structures we get are functionally relevant.
When we collect the genetic interaction data, it's not just for the sake of determining the structure. We're going to be learning a lot of the biology and the different pathways that certain proteins are involved in the cell. So it's two things that we're going to be learning at the same time by collecting this data, and we're going to be able to study systems that maybe have been refractory to structural biology for many years.
I think this is a great example of how you should never give up and never give up on something that you think is very exciting. And I've never had a paper take 15 years, but it's incredibly satisfying to see this come to fruition and get it published. And I think it's going to have a big impact in the world of genetics and structural biology, and putting these together to help understand how different mutations result in different disease areas.